Okay, so today we're going to be covering chapter 28, where we will look at invertebrates. So to define the term invertebrate, this is going to include all animals that lack a cranium, which is a skull case, right? A bony casing around the skull, around the brain. So you could say cranium or, again, skull case. And they also lack a defined vertebral column or spine. Okay. Most invertebrates also lack an endoskeleton. However, some have an endoskeleton. Like, for example, in the picture down here of the sea star, the starfish, which is an echinoderm, actually does have an endoskeleton. Okay, so there are some exceptions to that rule. The other group of animals that we're unfortunately not going to probably have time to talk too much about are the vertebrates. Okay, and those would be animals that do have a cranium and do have a defined vertebral column. Many invertebrates are aquatic, however, some are terrestrial. So aquatic means that they're going to live in water, either marine habitats or freshwater habitats. And then some are terrestrial, which means that they are land dwelling. Okay, so back to this figure that we've already introduced you to in chapter 27. This is, again, our phylogenetic tree or cladogram showing animal evolution. And so on this figure, you can see that, I'm having trouble with my pen here. Again, so sponges, cnidaria, ctenophores, the platyhelminths or flatworms, rotifers, mollusks or bivalves, annelids, the lophophorites, the nematarians, the nematodes. We're not going to talk about all of these groups, but some of them we are. The nematodes we are, the arthropods we are, the echinoderms we are. And so all of those that I just mentioned are all considered to be invertebrates. When we get over here to the chordates, these guys are going to be vertebrates. Okay, and the next chapter covers vertebrates. Okay, so let me just back up one. We're going to start off right here at the beginning of our tree. The first animals to have evolved from the choanoflagellate ancestor, they're going to be multicellular, which is marked off right here. So our first clade or group are going to be from the phylum Porifera, or the sponges. So that's who we're going to look at first. Okay, so again, sponges, or more scientifically, the phylum Porifera. These are going to be the simplest invertebrates, and they are parazoan, so they have true tissues. Sorry, they are multicellular, but they lack true tissues. So that term parazoa means that they lack true tissues. Everybody else beyond the sponges, beyond the phylum Porifera, will be eumetazoan, which means that they have true tissues. Okay, so all organisms in the phylum Porifera are invertebrates that are multicellular, but that lack true tissues. So they don't have, organs or organ systems. But they are multicellular, and they do have specialized cells in their body. We'll talk about some of those. Okay, so again, we're talking about the phylum Porifera. That includes the sponges. These are going to be found in aquatic environments, mainly marine environments, and they're going to be found primarily in warmer waters. There are about 10,000 species that have been classified, and they range in size from just a few millimeters to more than a meter in height. Some species have also been found to form symbiotic, mutualistic relationships with bacteria or algae. So again, as stated here, these guys are going to be multicellular, but they will lack true tissues, which makes them parazoa, not eumetazoa, parazoa. And again, their cells do show specialization. We'll talk about the role of some of the cells in the sponge body on the upcoming slides. The sponge does go through a life cycle where it will start off as a larval form that is free swimming. So it's motile. And then along development, that motile juvenile larval form will settle down onto some type of a strata, and it will then attach itself to that surface and grow into the adult form, which we say the adult form is sessile. And so this term right here, sessile, means that it's not capable of movement. Okay, however, there is some recent data that suggests that sponges may actually creep a little bit. This has only been demonstrated in a laboratory setting, not in the wild. And when I say creep, sponges have been shown to move, a few millimeters over, hours of time. So it would be an extremely slow movement. However, it's still movement. And so, again, recent data is coming out that the adult may not be completely sessile. Okay, so morphology of sponges in the phylum Porifera. Sponges are going to be asymmetrical, so we cannot consider them to be radial or bilateral in symmetry. So we say they are asymmetrical. They don't really have true symmetry. They are cylindrical in shape, and they're going to be hollowed out in the middle, and that middle section is called a central cavity, or the scientific term for that is the spongiocele. So that's this term right here, the spongiocele. Oop, I lost my pen. Okay, so the spongiocele is going to be the inside center cavity. You're right inside of here, and here are some other structures here, and inside of here, right? All of those would be the spongiocele. If you look at the body wall, it looks like it has holes through it, and it does. Those are pores. The scientific name for those pores is ostia, okay? And they are incurrent pores, which means that water is brought in through those pores into the spongiocele, into that central body cavity. And that's really, really important because that's actually how the sponge feeds. They're filter feeders. So as water is pulled in through these pores, the cells of the body wall are going to pull out food. They're going to capture food and take it up. And then the water passes through into that central cavity. And then it's actually going to be, it's going to leave out of the top. The top of that cavity, that hole there, is called the osculum. Okay, that's that term right here, osculum. Again, that's a pretty large opening, and it's usually on the upper or top surface of the sponge. Okay, so this is a nice picture. It's showing, a cartoon image of the sponge that you just saw in the last slide. But what we've done is we've taken part of the body wall and removed it so that you can actually see the inside. You can see that the inside here is hollowed out, right? Is hollowed out. Let me grab my pen so it doesn't do that. And so again, that hollowed out region in here is called the spongiocele. And so again, water is going to be pulled in through these pores called ostia. And as it's being pulled in, food is going to be filtered from it. And when that water gets in here, it's then going to be expelled out of this top portion, the osculum, the hole at the very top here. Okay, so it's one-way flow of water in through the pores or ostia, into the spongiocele, and then out through the osculum. Now let's look at the body wall. In this figure, it's a little bit better. We can actually see up close, so we've taken just a segment of the body wall, maybe right here, and we've magnified it. Okay, so we can really see what's there. Okay, and so, let me just make sure I have my pen here. If we magnify that section, oh, I circled it, but it's gone, section, and we kind of blow that up or magnify it, what we see here are some different cell types. And so the outermost cells here, in pink, okay, there's a layer there. They are kind of like the skin or the epithelium of the sponge, and they're going to be made up of cells here. We don't call them epithelial cells. Instead, we call them pinacocytes. Okay, they are epithelial-like, so they're going to cover the surface the outer surface of the sponge. And then we have an inner, this yellow region in here, that's going to be a layer of jelly-like material called the mesal. Okay, and all of that mesal, all of that jelly-like material has been secreted by cells, so it's actually part of the extracellular matrix. And that layer is going to act like an endoskeleton, but it's not hard. It's going to help to maintain shape. There are hard structures that we may find called spicules. So do you see this structure here? Looks kind of like a spine. That's a spicule, and it's believed that those are there to help the body maintain shape. Okay, so between that and the mesal, that jelly-like material, that's what really holds this organism, kind of together and provides some structural support. We don't really have bones here. Instead we have these structures. Again, these are in aquatic environments, and so there's less need for structural support structures in the body, but still they're there, okay, to help to maintain shape. Okay, what else? So those ostia, those pores, that's being shown right here. This is a pore going straight through here. Ooh. And you'll see that there's actually a cell, this purple cell, this is the nucleus of that cell, that lines that structure sometimes. And if that cell is there, that cell is called a porocyte. Okay, so a porocyte is going to be the cell that lines the pore. We have a couple of other cells there. This is a figure from your book. It's the same thing. So here we've got, my pen keeps going away. Here we've got our basic sponge body plan, right? And so here, what we've done is we've really sectioned through it. And so you're looking at the sponge from the side. And so again, right here, these cells, that's actually blown up over here. So these cells on the very outside, again, those are those epithelial-like cells called pinacocytes. And again, they're going to form that outer covering of the sponge, kind of like the skin of the sponge. Okay, what else did we already talk about? We already talked about the porocytes, right? So those are going to be cells that line the pores. It controls water flow through those pores, or those ostia. We did talk about spicules. There's one right here. We talked about the mesal. Okay, now there's some other cells in here that we haven't talked about yet. There's only just a couple more that I really want to go through. The first one are called the choanocytes, or collar cells. We actually mentioned these back in chapter 27. We said that these cells really resemble the common ancestor of all animals, that choanoflagellate, right? And so these cells look very similar to that. Those cells are right here. Okay, this line of cells right here. So the nucleus will be in here, and then we've got this long whip-like tail called the flagellum. And then you'll notice this structure here. This is the collar. Okay, and that collar is going to be an extension of the plasma membrane, and it's going to form like a ring, a collar, around the flagellum. These cells are really, really important in the sponge. They do a couple of things. The first is they generate the current. Okay, so what does that mean? These whip-like tails, they're going to beat, and as they beat, that's going to cause water to be pulled in through the ostia. Okay, so they are going to be responsible for creating a current that draws water in through those pores. So why is that so important? Because again, that's going to draw in food. So with the water comes food. These collar cells, choanocytes, also are going to filter food out of that water. So they're going to be responsible for trapping, and then they also ingest food particles. The collar is believed to work like a sieve, and so water can pass through and get in, go past these cells and go into the cavity of the sponge and then out through the top. But any food particles that are coming through, they're going to get trapped on that sieve. Okay, so really, really important for feeding. These are kind of neat too. They will differentiate into sperm for sexual reproduction. Okay, so they not only are responsible for filter feeding, but they will also differentiate and form sperm cells that will be used in sexual reproduction. So very, very important cells. I want to mention one other type of cell, the amoebocytes. And they're shown here in green. And the amoebocytes are going to be found in that jelly-like material, again called the meso. And their job is to deliver nutrients from choanocytes. So nutrients will diffuse into the amoebocytes from the choanocytes. And the amoebocytes actually can move throughout the meso, and they're going to make sure that other cells get nutrition. And so they're going to take nutrition from the choanocytes and deliver those nutrient molecules to other cells. Okay. Amoebocytes can also differentiate into other cells, like, for example, eggs. So right here in the meso, this other green cell is called the oocyte, or in layman's term, we would just call that an egg. Okay, so pretty neat. Okay, so here's some more information about sponges. So they lack a complete digestive tract. They don't have digestive organs. Really, the amoebocytes do all the food capture and digestion. Okay, so they really do lack complexity there. They also lack a circulatory system. Okay, so nutrients that are digested in amoebocytes, they're not going to circulate through vessels like we see in organisms like us. Instead, they're going to just diffuse from one cell type to another. So very, very simple. They also lack a reproductive system, right? So there's not specialized organs that produce egg and sperm. Instead, again, sperm will arise from choanocytes and eggs will arise from amoebocytes. They also lack a nervous system. So notice how we didn't talk about nervous tissue at all. No neurons, no brain, no spinal cord, none of those structures. Okay. They do not have a respiratory system. Okay, so gas exchange, circulation of those gases, excretion, all of those processes are done by basic diffusion. Okay, pretty simple. Digestion is intercellular. So sometimes students, here we go. Sometimes students will get a little bit confused as we go on and look at other animals that have digestive structures in the center that kind of look like sponges. Sometimes they'll get a little bit confused. So I just want to make sure we point out that digestion does happen inside of cells, right? Not in a cavity or a gut. It's actually happening inside of those choanocytes. Okay, and again, their mode of nutrition, we call them filter feeders. These guys will reproduce both sexually and asexually. So sexually, what will happen is either fragmentation, where a piece of that sponge literally just kind of falls off or breaks off, lands somewhere and starts to grow into a new organism, or budding. That's when a specialized structure, a polyp, grows out of that sponge and then buds off. Okay, so those are the two types, both, again, asexual. Sexual reproduction, again, you're going to have an egg and sperm. So gametes are produced. And sponges are going to be hermaphroditic. So that means that they can produce both egg as well as sperm in the same animal. Sperm get shed into water. Those sperm will then swim to a neighboring sponge and will enter the sponge's body and will fertilize the egg. The egg is going to be inside of the meso. And then after fertilization, you end up with a zygote, and that zygote will develop into an embryo, which eventually develops into a larval form, and that larval form will be released from the parent sponge, and it's free swimming. And again, it will eventually settle down and grow into the adult sessile form. Okay, so take a break. Here's your first Are You With Me? So you want to describe what an invertebrate animal is, where we most likely will find them, And then you can talk specifically about the phylum Periphery. You're going to describe key features. So for example, the morphology, what type of feeding and digestion, and what is reproduction like. Now I tell you to make a sketch and label the parts. And if you can do that, fantastic. If you are unsure of how to upload a picture, you can always just describe this. Either way is fine. So you want to talk about the different cell types, what the body wall looks like, and make sure you use proper scientific terminology.
Okay, before you go on, I have a couple of questions for you, just like usual. I know I just said that if you can't sketch, you can describe, but you guys know how to sketch and you know how to upload. You've become pros at that. So go ahead and sketch the structure of the periphery and label and describe the parts as well. You can do all of that. Okay, so here's a couple of questions. The first one, the large central cavity of the sponge is called is it the osculum, the spongiceal, the mesal, or ostia? The large central cavity. So that is called the spongiceal. Remember, if you chose osculum, the osculum is the opening at the top of the sponge. Okay, that's that large, usually large opening. But the actual cavity itself is called the spongiceal. Okay, and the rest of those don't make sense. Okay, next question. All of the following are true of the phylum Porifera, except, are they multicellular? Are they filter feeders? Can they reproduce both sexually as well as asexually? Do they have true tissues? So which of those is not true? Give you a minute to think about that. Okay, so hopefully you chose yellow. Okay, the Porifera do not have true tissues. They are multicellular. They do show specialization of cells, right? So certain cells have certain jobs, but they don't have true tissues. So there's not cell types that form a tissue and tissues that form organs and so on and so forth. Okay, and the rest of those are correct. So again, the answer there would be yellow. The phylum Porifera lacks true tissues. Next question. This cell type is found in the mesal. It delivers nutrients, and it gives rise to eggs. Are those pinacocytes, porocytes, amoebocytes, or sclerocytes? So which one of those is found in the mesal, is responsible for delivering nutrients to other cells, and gives rise to eggs? Okay, so hopefully you answered amoebocytes. Okay, all of those functions describe what the amoebocytes will do. Last question, I think. Yeah, last question. This cell type creates a current and traps and ingests food. Are those choanocytes, pinacocytes, porocytes, or amoebocytes? So which one of those is responsible for creating a current, an incoming current of that water, and for filtering out food, so trapping and ingesting food? Which one of those cell types? Hopefully you answered choanocytes. Remember, those are those collar cells. They've got those whip-like tails called flagella. Those flagella are going to move, and that's going to draw water in through those pores, or ostia. And as that water moves through, the choanocyte, it's really the collar, is going to trap food particles and then internalize those or ingest them. And that they're going to really be supplying nutrients to the rest of the cells of the sponge body, with the help of amoebocytes. Okay, so the actual cells that create the current, trap, and ingest food are the choanocytes. Again, we commonly call them collar cells. Okay, so you're going to go on to the next section, and then I'll be back with some more Are You With Me questions.
Okay, so here we are back at our phylogenetic tree. So we've tackled the sponges so far. And so the next place we're going is going to be the cnidarians. Okay, and so the cnidarians are going to be different from the sponges in many ways, but the most prominent way is that they will have true tissues. So they are multicellular, but they are also eumetazoa, which again means that they have true tissues. Okay, so what else? What other features do we have for this phylum Cnidaria? So animals that belong to this phylum are going to have radial symmetry. So remember back to chapter 27, we talked about what radial symmetry is. I always picture kind of a can of soup. If you look down at it from the top, right, you can kind of cut that can of soup along multiple axes to get mirror images. So the body plan really is radial. It radiates around that central axis. Okay, very different from a bilateral animal where there's only one, division that you could make to get mirror images. Also with radially symmetrical animals, you don't have a right and left side, right? But you do have a top and bottom. Okay, you also don't have a front and back, just a top and bottom. So organisms belonging to Cnidaria, they're going to be radially symmetrical, and they are also going to be diploblastic. So remember, these are going to be animals that have two embryonic germ layers. They're going to just have ectoderm and endoderm. They're going to lack mesoderm. Okay, and these are going to be mostly marine animals, definitely aquatic, mostly marine, and they're going to contain specialized cells that are going to contain these really cool stingers. So the cells are called cnididocytes. Let me grab my pen here again. It keeps shutting off. The cells are called cnididocytes, which again, in layman's term, those are called stinging cells. So all animals belonging to the phylum Cnidaria have these. And inside of these cnididocytes, inside of these stinging cells, there's going to be a structure called a nematocyst, which again, in layman's terms, is a stinger. Okay. These are going to be found around the mouth, and they will also be found at the end of the tentacles. And so what are they there for? So Cnidaria are not filter feeders. Instead, they're going to be active predators. And so these stingers are going to contain toxins. And when these stingers are released, there's a trigger that actually releases them. They're going to stick into prey, right? Some unlucky animal passing by. And the prey will be immobilized by the toxins inside of these tentacles. And then the Cnidaria, whatever kind of species it is, will take that prey and they'll actually they'll stuff it right into their gastrovascular cavity. We'll take a look at that. This is what those cells look like. So you can see all these thread-like structures. Those are all tentacles, okay? And at the ends of those tentacles, they're really tiny. You can't see them so well. There are going to be these cells, okay? And so these cells are called, again, cnididocytes. Oops, sorry. Cnididocytes. And again, the nematocyst, the actual stinger, is inside of here right now. It hasn't been discharged yet. Here's the trigger right here, or the cnidocil. Okay, and that trigger, it can be activated because of pressure or chemical. There's multiple types of stimuli that can cause that trigger to release the nematocyst. Once the nematocyst is released, at the end of this, there's this. Right, so there's a thread, and then look, there's these barbs, and inside of here, there's oftentimes, again, toxins. Okay, and so once this sticks into the prey, toxins will be released, and the prey becomes immobilized, and then those tentacles can just pull that prey right in. Okay, so Cnidarians will come in two distinct morphologies, and some species alternate between these, and then some of them do not. So within one life cycle, both of these might be displayed. So we've got two forms. So we'll do the polyp form first. So the polyp form is going to be sessile. So this is the base of it, or the bottom, and that's going to be attached to some surface, and then this would be the top here. Okay, now the top of the polyp form is going to be where the mouth is, or I should say the opening, right? This is where food is brought in. Okay, and these structures here would be part of the tentacles. Okay, you've seen these last week in lab. You've seen a couple of different species here, pictures of them at least. You saw Hydra, you saw Obelia, okay? You also saw Metridium. Those are all examples of the polyp or stalk stages of those species, okay? Again, the mouth is on top, or I should say the opening to the digestive system is on the very top there. And again, tentacles will surround it. So that's the polyp. The medusal stage, or sometimes we hear it referred to as the bell stage, okay? It's going to be very different. It's going to be free swimming, right? Not sessile, instead free swimming. It's capable of movement. And if we look at this, this would be the top side, and down here is the bottom side. This is actually the mouth, or the opening to the digestive cavity. And tentacles will be on this side, on the bottom side. Okay, they'll be hanging down. We oftentimes refer to these as jellyfish, right? And so in lab, the one that you saw was Aurelia. Okay, that was our example there. Okay, so again, radially symmetrical, diploblastic. You can actually see in blue here, well, we're going to name this, but this is tissue derived from endoderm. And in red, we've got tissue derived from ectoderm. And the gray in between them is not mesoderm. Instead, it's a jelly-like substance, which we're going to name mesoglia. Okay, so they are diploblastic. Again, some species will display both morphologies during their life cycle, and some just one. It depends on the species. Okay. Here's just a figure showing some of these. This is the polyp stage over here. And these guys are much more complex. They have, a basic body form where they'll have some structures that are coming off that are used in feeding, but some are used in reproduction. So there's different structures here. This is a cartoon. Obviously, here's another cartoon. It's a little bit better. This shows the life cycle. This particular life cycle is Obelia, which again, you saw in lab. You particularly saw this structure here in lab, a polyp. Okay, so let's look at this life cycle. So we'll start with the polyp. So this is a polyp colony. You've got kind of multiple polyps here. And what we have are some different structures. So if we go to the top structure here, this is a feeding polyp. This is going to be used for eating. So the mouth is here, and it's surrounded by tentacles, which are going to be used to grab prey. The same thing here. Here's another feeding polyp. So again, we have the mouth here, tentacles, and this inside compartment, the gut, is called the gastrovascular cavity. And we'll take a look at the body in just a second. But we also have other structures that are not used for feeding. This one here is a reproductive polyp. And so this polyp will actually develop into the medusal stage. Okay, so the jellyfish stage. And so here you can see we've got two medusal jellyfish, right? These have both been produced by the polyp colony. And there will be male and female medusa will produce egg, and that gets shed into the water, and male will produce sperm. Those get shed into the water. If sperm and egg come together, you get fertilization, and that will result in a larval form called the planula larva. All of this here is free swimming. The planula larva will settle down, and it will develop into a polyp, which is, again, sessile, non-motile. Okay, so again, some from the phylum Cnidaria do have life cycles very similar to this, and then some will only be medusal, and some will only be polyp. Depends on the species. Okay, so let's take a look at the morphology a little bit more. So again, the outer body wall is going to be called the epidermis, and that develops from the embryonic germ layer called ectoderm. Okay, so we've got the epidermis on the outside that is derived from ectoderm. On the inside, we've got a layer called the gastrodermis, which develops from endoderm. And that gastrodermis is going to be the inner lining of the gastrovascular cavity, or in short, we can call it the digestive cavity. Okay? And then in between these two layers, we're going to have a non-living jelly-like layer called the mesoglia. And again, the role of that layer is to provide structural support. It's acellular. The contents of the mesoglia has all been produced by cells, either of the ecto or the endoderm. Okay, what else? So members of the phylum Cnidaria are going to, again, have differentiated cells that form tissues. They are going to have nerve cells. They do have contractile epithelial cells. So when we think about contraction, movement, we're kind of thinking about muscle, right? Not true muscles, but cells that do show some movement, some contraction. They have enzyme secreting cells. Okay, so they don't have specialized digestive structures, but they do secrete enzymes. So they also have nutrient absorbing cells. We don't really say that they have true tissues. Sorry, they do have true tissues. They don't have true organs or organ systems, but again, they have specialized cells that form tissues. Okay, so we see that the beginnings of complexity in these organisms. Let's talk about digestion. So digestion is internal, so it's inside the animal's body, but it's not in the animal's cells. So we say that it's internal but extracellular. So food is brought in through the mouth via tentacles, and then that food will pass, be shoved in, pass through the mouth into the gut, which in scientific terms, again, is called the gastrovascular cavity. Okay. Digestion is going to happen in that structure, not inside of any cells, but inside that structure. The cells lining the gastrovascular cavity, so those are going to be gastrodermal cells. They're going to secrete digestive enzymes. That's going to allow that food to be broken down, and the nutrients will be absorbed by those cells. So again, by those gastrodermal cells. They don't have any, they don't have a respiratory or a circulatory system per se. Instead, nutrients and gas exchange just occurs via diffusion. Okay, so it's just simple diffusion. Okay, let's talk about some of the different classes. So again, there's about 10,000 species that have been identified in this phylum Cnidaria. And they get split into 4 classes. And those classes are the Anthozoa, the Scyphozoa, the Cubozoa, and the Hydrozoa. And we're just going to briefly look at these classes. So the class Anthozoa first, we saw one of these in lab, if you remember, Metridium. Okay, one of those pictures or that you looked at, or the organism that you looked at. So organisms that belong to this class Anthozoa, they're going to be found in the polyp stage only. So they do not have a medusal stage. That's going to include sea anemones, which Metridium was a sea anemone, sea pens, and corals. There's about 6,100 species, so there's quite a few of these. Okay, and you can see the body shape is just as we described. So you've got a mouth here surrounded by tentacles, right? There's an epidermis, mesoglia in between, and then that gastrodermis on the inside. And this inside structure right here is that gastrovascular cavity. This is where digestion occurs. And then nutrients are absorbed through the body wall here, through those cells. Any waste that is produced will actually go right back out of the top. Okay, so there's also no specialized excretory structures. The mouth is going to serve as both an opening to bring stuff in as well as to get rid of waste. Okay, the Scyphozoa is the next group. The Scyphozoa are the common jellyfish. So the one that you saw in lab representing this particular class was Aurelia. Okay, these are marine animals, so found in aquatic marine environments. There's only about 200 known species. And again, the medusal stage is prominent. Some of these will go through a life cycle where they do have a polyp stage as well, but the medusal stage is the longer lived, most prominent stage. As far as size, these guys vary from 2 to 40 centimeters in length. Okay, so they can be pretty teeny. However, it has been recorded that there is one species, and you can look this up online, that has been recorded to be about 2 meters in diameter. Well, that's pretty big. Okay. If you take a look at the medusal stage here, this guy's beautiful. So this would be the top surface, the upper surface. And on the bottom surface here is where the mouth would be. Okay. And if we want to use better terms here, the surface that does not have the mouth is called the aboral surface. And the bottom surface here that does have the mouth is called the oral surface. Okay, or you could just say top and bottom. On this bottom surface, the oral side, again, you're going to have the mouth, and you can see all these structures hanging down here. Those are all tentacles. And those tentacles, again, are going to have cnididocytes that bear nematocysts. So they're going to be used to sting prey, paralyze them, and then that prey will be pushed into the mouth area. And that mouth area is going to lead to the gastrovascular cavity. Digestion will occur, nutrients get absorbed, and then any waste products come right back out the mouth. Okay, which is right here. So notice how it says mouth/anus. One opening there. Okay, so not a complete digestive tract. Okay. So again, just wanted to show you a life cycle from this class Scyphozoa. So even though the medusal stage is prominent, some of them will go through a polyp stage as well. So again, you have egg and sperm shed into the water. Fertilization happens, you get a zygote. That zygote develops into a larval stage, still free swimming. That larval stage will eventually settle down and develop into the polyp. This is a mature polyp, right? And that polyp will produce medusal buds or reproductive buds, which will give rise to the free swimming medusal stage. Okay. Okay, the next class are the Cubozoa. These are called the box jellies. These are pretty tiny. They range from 14 to 25 centimeters in length. You can actually see there's one inside of this little tube here. Look how teeny he is. Okay, only a few centimeters in length. They're called Cubozoa or box jellies because of their shape. They're kind of square, at least on their undersurface, they look really square. And they have tentacles that come off of them from their 4 corners. Okay, so there's a collection of tentacles in 4 locations or the 4 corners. These guys are the most venomous group of jellyfish. So really, really dangerous. If you get stung by one of these guys, it can be deadly. Okay. All right. The next class, the last class here, is the class Hydrozoa. And again, you saw a couple of these. You saw Hydra and you saw Obelia. Both of those belong to the class Hydrozoa. There's about 3,200 species here, so a pretty big group. Again, mostly marine, all aquatic, mostly marine. And they're going to show complete life cycles where they exhibit both the polyp and the medusal body form. Okay, this is Hydra here. You can see the main body structure here, and then here's a bud. That might be a reproductive bud or a feeding bud. You can't tell just by looking at it. And so, again, the same structures. So the outermost layer of cells is still going to be called the epidermis. That's derived from ectoderm. The innermost layer of cells is going to be called the gastrodermis, derived from endoderm. In the middle, you're going to find that jelly-like substance called the mesoglea. On the very inside of this organism will be the gastrovascular cavity or the gut. And at the top here, you have your mouth. And that mouth will be surrounded by tentacles. And those tentacles will be lined with stinging cells, nidinocytes containing nematocysts. Okay, so the same kind of thing here. Okay, so that's it for the phylum Cnidaria. We're going to go on to the phylum Ctenophore before we have our next Are You With Me? The phylum Ctenophore, these guys are also, they're jellyfish. They look like jellyfish. They are going to be bioluminescent, so they're going to glow, and they're marine animals. Okay, they're different from the Cnidaria, and there's a couple of different features here. First off, they're going to have these rows. You see these rows of cilia. Okay, and those rows of cilia, they look like combs, and so that's where they get their name from, comb jellies. And the beating of those cilia is precisely controlled by this organism, which is kind of neat. The beating of the cilia is actually controlled by a very primitive nervous system. It's really a nerve cell network. These guys also have some other, sensory type organs. The sensory type organ that's going to be responsible for this controlled beating is called a statocyst. So there is, again, kind of a primitive nerve system. There is this nerve cell network and specialized sensory organs. Pretty neat. So what did we cover so far? So we said that there's 8 rows of cilia that resemble combs. Their beating is going to be controlled by nervous tissue. Again, they are radially symmetrical, just like the Cnidaria. They are diploblastic, so 2 cell layers, and those 2 cell layers are separated by mesoglia, so that's kind of similar. They do have tentacles that surround the mouth. The tentacles here in this phylum, they do not have stinging cells. Instead, the tentacles have this glue-like substance. They're adhesive. We call them glue cells. So when a tentacle reaches out and sticks to prey, the prey can't get away. It's literally glued to the tentacle. The tentacle will just pull that prey back in. Okay, so pretty neat. What else? So another advance that we see in the phylum Ctenophore is their digestive cavity has 2 openings at both ends. So it's going to have, 2 openings in total, 1 at both ends. It's going to have a mouth where food enters into, and that food will be digested in that gut. Nutrients are taken up, and then any waste products are going to leave via a different opening, the anus. So if we look here in this figure, the mouth is going to be here on the underside. That will be the oral surface. And the anus will be on the other side here, on the aboral surface. So a little bit more advanced. Okay, so here's your second Are You With Me? You want to name some key features in the phylum Cnidaria. Tell me about nidinocytes and nematocysts. Tell me about the life cycle. What's a polyp? What's a medusa? What do they look like? Sketch them or describe them. How does digestion take place? Do these organisms have organs and organ systems? And then tell me just a little bit about the 4 classes. And then key features of the phylum Ctenophore. What do they look like? How are they different from the phylum Cnidaria? Once you're finished with those, you can continue on.
Okay, so before we go on, another couple questions for you. Okay, so here's the first one. All of the following describe the phylum Cnidaria except, are they eumetazoan? Do they display radial symmetry? Are they filter feeders? Do they contain nidinocytes with nematocysts? So which one of those is not true of the phylum Cnidaria? Okay, so hopefully you answered filter feeders. Okay, so the Cnidaria, the jellyfish, the sea anemone, they are not going to be filter feeders like the sponges, right? They're going to be, carnivorous predators. Okay, so those tentacles with those stinging cells will be used to capture prey. And then that prey will be stuffed right into the mouth. They only have that single opening into the mouth. and digestion is going to happen right inside that gastrovascular cavity. Okay, so they are definitely not filter feeders. They're not pulling food out of water that's being brought in like the sponges. The rest of those are true, so let's make sure we understand these terms. So when we say eumetazoan, that means that they have true tissues, and so yes, the Cnidaria do have true tissue. Everybody on our cladogram, except for the peripheral, the sponges, everyone else is eumetazoan, having true tissues. These guys, that's my husband in the background, these guys do display radial symmetry. Right, they are not bilateral. They are definitely going to be radially symmetrical. And again, lastly, they do contain nidicites. Those are those stinging cells. Okay? So again, the one that is incorrect here is that they are not filter feeders. Okay, next question. A scientist finds a radially symmetrical diploblastic organism. The organism displays the medusa body form. Maybe I should say medusal. Anyways, body form. Has openings at both ends of its digestive tract. So it has two openings, not just one. And has tentacles that are sticky. Which phylum does this organism most likely belong to? Is this an organism from the phylum Porifera, the phylum Cnidaria, the phylum Ctenophora, or the phylum Nematoda? Okay, so if you need longer, just pause me. Hopefully you answered green. This organism most likely belongs to the phylum Ctenophora. So what gave that away? Well, radially symmetrical, diploblastic, medusal body form could be either the Cnidaria or the Ctenophora. Okay, so the phylum Porifera and the nematodes, which we haven't talked about in too much detail yet, those two would automatically be out. So you're left with Cnidaria and Ctenophora. So what gives it away that it's the Ctenophore? Two things here. The fact that they have an opening on both ends of their digestive tract, okay? The Cnidaria only have a single opening, where the Ctenophores will have two openings. And the second, it says that its tentacles are sticky. So remember that in the phylum Ctenophora, we don't have nidicites with nematocysts. Instead, we have glue-like substance at the ends of these tentacles. Okay, so it says the tentacles are sticky, so that gives you the impression of a glue-like substance. So again, hopefully you answered green, Ctenophore. Last two questions for you. All of the following describe Cnidarian nutrition or digestion except: are they heterotrophic? Are they carnivorous? Does digestion take place internally? Does digestion take place intracellularly? Okay, so think about that for a few minutes. Again, if you need more time, just pause me. That's the great thing about having these videos. Okay, so Cnidarians, yes, they are heterotrophic because they don't make their own food, right? So that the first one makes sense. The second one, they're carnivorous, yes. Okay, they're going to be carnivorous. They're going to trap their prey, again, stuff it into that gastrovascular cavity. These next two are a little bit tricky. So the first one says internal. So is digestion internal? Does it take place inside the organism's body? And that's going to be a yes. Okay? Unlike organisms that we've already studied, for example, fungi. Remember we said that fungi, their digestion is going to be extracellular. So what a fungus does, fungal cells will secrete digestive enzymes into whatever they're growing on. Those digestive enzymes will complete digestion outside of the organism's body, and then the fungal cells will then absorb those nutrient materials. They'll take it up, either by active transport or diffusion, whatever. So in the fungi, digestion takes place outside of the fungal body, where in the Cnidarians, the digestion is going to happen inside of the gastrovascular cavity, which is a compartment within the body wall of the organism. So yes, digestion is internal. Now the last one on here, yellow, it says intracellularly, so that means inside of cells. Okay, now that doesn't happen. If you remember back to the sponges that we talked about in the previous section, sponges had intracellular digestion, right? Digestion was happening inside of those choanocytes. Okay, here it's different. Digestion happens in the gastrovascular cavity, which is a cavity, a hollowed out space, but it does not happen inside the cells. After digestion is complete, it's going to be those cells that line the gastrovascular cavity that are going to take up those nutrients and then distribute it to the rest of the cnidarian body. Okay, so the one that is not true in this list is yellow, intracellular. Okay, digestion is internal but extracellular, inside the gut, not inside of cells. Okay, last question. The sessile form of members of the phylum Cnidarian is called so which of these is the sessile form? Is it the polyp, the medusa, the bivalve, or the mesoglea? Which of those is the sessile form? Okay, and hopefully you answered here the first one, red or pink, the polyp form. So polyps are going to be sessile. What does that mean? It means that they pretty much are going to be attached to some substrate, whatever they're growing on, and they're pretty much not going to move. Okay, their tentacles will move, okay, but they won't actually have a change in location. Okay, so they are sessile. They kind of sit in one spot and really don't move from there. Where the medusal form in blue, that's the complete opposite, right? That's the free swimming form, which is not going to be attached to any substrate. Instead, it's going to definitely move throughout the water. Okay, so that term sessile, kind of lacking in movement, stationary. So polyp is the answer there. Okay, so you're going to go on to the next section, and then I'll be back with some more questions.
Okay, so that brings us to the next group up. So we've completed the phylum. My pen. We've completed the phylum Porifera, which includes the sponges. We've completed the cnidarians and the ctenophores. And so again, everybody's multicellular. Only so far the cnidarians and the ctenophores have true tissues, and both of those are radially symmetrical and diploblastic. So now we're going to get into animals that show bilateral symmetry, that are triploblastic, and that either have or lack a coelom, a body cavity. Okay, and what we're going to talk about first in these bilateral animals, we're going to talk first about the protostomes, and we're going to focus our attention on the Lophotrochozoa first. And we're going to just talk about a few of these groups. We will talk about the flatworms, which are scientifically known as the platyhelminths. We'll talk briefly about the mollusks, and we're going to look at the annelids. I think that's it. Okay, so let's jump right in. Okay, so again, the Lophotrochozoa, these are going to be multicellular animals that have true tissues that are bilaterally symmetrical. So again, if we think about a bilateral animal, there's only going to be one plane, right, that sagittal plane, that we could make a cut to yield right and left sides or mirror images. So bilateral animals have a right and left side. They also have a dorsal and ventral surface, and they also have an anterior and posterior end. And with bilateral symmetry, we also see cephalization. So that second term here, we talked about that in chapter 27, but just to remind you, cephalization is going to be the evolution of a region, the anterior end, where there is a concentration of nervous tissue. So think about a brain or a head region. Okay. These bilateral animals are also triploblastic, which means again that they have 3 embryonic germ layers. So they have both, are all 3, so ectoderm, endoderm, and mesoderm. The Lophotrochozoa are also divided into the protostomes. And we had protostomes and deuterostomes. We talked about the differences between the two back in chapter 27. So you can kind of look back at that. Remember that there was a difference between early embryonic cleavage. So remember that the protostomes showed spiral cleavage. That cleavage is determinate, right? And we also talked about the differences between the formation of the coelom. Okay, so again, you can kind of check back at chapter 27 for those. What else do the Lophotrochozoa possess? They possess something called a lophophore at some point in their life cycle. So what is that? The lophophore is going to be a ring of ciliated tentacles. Okay, so tentacles that have hair-like structures. And that structure is going to be found surrounding the mouth, again, at some point during the life cycle of these organisms. Okay, we'll find the lophophore. The 3 groups, the 3 phylum that I'd like to talk about are the platyhelminths, or again, the flatworms. We'll talk about the mollusks, and we'll talk about the annelids. Those were the 3 groups that we talked about in lab, so we'll focus on those. Your textbook does talk about the other phylum, but you won't be held responsible for those. Okay. So our first phylum, the phylum Platyhelminths, again, the common name for these are the flatworms. These guys are acoelomates, which means that they do not have a fluid-filled body cavity. Okay, instead their bodies are pretty solid. They do have a gastrovascular cavity. We'll talk about that in just a second. So they're acoelomates, which again means that they don't have a fluid-filled body cavity. They may be, they may be free-living, or they may be parasitic. Okay, so free-living would mean living on their own, either in the dirt or in an aquatic environment, so either terrestrial or an aquatic environment. But parasitic means that they're going to be living on or inside of another organism. And so we find flatworms in both cases. So some are free-living and some are parasitic. A common feature shared by all flatworms is that they have a gastrovascular cavity, right? We already saw something like that when we looked at the phylum Cnidaria and Ctenophore. So they have a gastrovascular cavity. It's incomplete in that they only have one opening, usually. Okay, so the mouth where food is brought in will also serve as the anus. Although some do have a separate anus. Okay, it depends on the species, but most have only one opening. Digestion happens internally inside that gastrovascular cavity, but again, extracellular. So not inside of the cells, instead inside of the cavity. And so digestive enzymes will be secreted from those from those cells lining the gastrovascular cavity, and then digestion is going to happen right inside of there. Excretion depends. In some species, there's going to be these specialized cells called flame cells, or more scientifically known as nephridia. And so body waste will sometimes be excreted through those cells, which are usually on the belly side or the ventral surface of the organism. I'll show you a picture that has some of those in them. So yeah, they have, they do have a digestive system. They also have a primitive nervous system. The nervous system is going to consist of a structure called the ganglion, which is kind of a primitive brain, and that's going to be at the anterior or head end of the organism. So there's a concentration of nervous tissue right there. And then we're going to see that there is going to be a pair of nerve cords that are going to run the entire length of the organism's body on both the right and left side. And these will be connected in the middle. And so what we end up getting is a structure that kind of looks like a ladder. It's pretty neat. I'll show you on the next slide what that looks like. They also can sometimes have some sensory cells or sensory organ structures. Some of those sensory organs might be photosensory, so they might respond to light, and some might respond to chemicals, so they might be chemosensory cells. And those are usually concentrated also at the anterior end, the head region. These guys do not have a circulatory system, so there's no specialized structure, no vessels, no primitive heart type structures. We don't find those. And there's also no respiratory structures. So we don't find gills or book lungs or any structures like that. And so nutrients as well as gases are going to just diffuse, by basic diffusion through the body wall and into cells. Most are hermaphroditic, so they will produce both egg as well as sperm. And fertilization here is internal, so we don't have the shedding, of egg and sperm cells. Instead, we have internal fertilization. Some of these guys can also reproduce asexually as well. Okay, so here's the basic structure. This particular platyhelminth in this figure is a planaria, which is one that you saw in lab. So he's a flatworm. Pretty neat. So he definitely has, let me get my pen, an anterior end and a posterior end. Okay. Let's take a look at these guys. So at the anterior end, the head region, we have a concentration of nervous tissue. So this guy's got eye spots. Okay, those would be photosensory organs. There's also a concentration of nervous tissue here called the cerebral ganglia. Okay, so again, a kind of a primitive brain. And then extending from the cerebral ganglia, we have these two nerve cords, one running on the right and one running on the left-hand side of the body. Those are called, again, those longitudinal nerve cords. Notice how they're connected in the middle. Okay, those are called transverse nerves. Don't worry too much about these names. But if you take a look, kind of tilt your head to the side here, do you see the ladder network that we have here? So again, in yellow is all the nervous tissue. So again, it's called a ladder type nervous system. Okay, pretty neat. What else do these guys have? They do have a primitive digestive system. And so it's kind of weird how they're showing this, the mouth and anus. It's on the ventral side. Here is the opening here, so just one opening. Food is going to be brought in to the gastrovascular cavity, or in this case, they're calling it the intestine. Okay, digestion will happen inside of this structure. Nutrients will be, diffused into other cells, and then waste is usually excreted out that same hole. So this serves as both the mouth as well as the anus. We do see some other excretory structures. So another way to get rid of waste here, running the length of, found along the length of the organism's body, these are those flame cells. So this is what that looks like here. And again, there's an excretory pore, which is going to allow waste materials to leave. Okay. What else? That's pretty much it on here. Okay, so pretty simple. There are 4 classes of platyhelminths, and we'll just kind of briefly talk about each of them. There's the class Turbellaria. This is shown here in this first picture, if you can see that guy here. Oops, sorry. These are shown here, so this guy here. These are aquatic, living in marine environments. Again, they're flat and cross-section, so they appear pretty flattened. They do have a top or a dorsal, as well as a belly side, which is the ventral surface. Okay, anterior, posterior region. Same structures that we saw in planaria on the previous slide. So again, they're going to be free-living, mainly marine animals. The next class, and we'll talk a little bit about some of these a little bit in more detail. The next class, these are called the Monogenia. These are going to be ectoparasites, mainly on fish. So these guys are parasitic, not free-living. An ectoparasite means that they're going to live on the outer surfaces of fish. And you can see this guy here. It's going to be a flatworm. This is the head region here. It's going to use the head region to attach to those fish. Trematodes. Okay, this is the third class. The trematodes are also called flukes, and you saw some of these in lab. Trying to think of the ones that you saw. You saw Fasciola. He's a fluke. That might be the only one that you saw for here. These guys are going to be parasitic. They're going to be internal parasites of animals, and they have a really complex, I like to say neat, life cycle that I'm going to talk to you a little bit more about on the next slide, so we'll go back to that. That's what some of these look like, okay? And again, the one that you saw a picture of was Fasciola. And the last class, which again, we'll talk about their life cycle too, because it's just kind of cool to see these, the class Cestoda, which is the tapeworms. And you saw a tapeworm in lab. Its scientific name is Tinea. Okay, so that's the one that you saw. These guys are also parasitic. Again, internal parasites. Very, very complex life cycle, just like the flukes. So again, we'll talk about both of those. Check out this tapeworm. Look how long he is. Pretty cool organism. Okay, so some platyhelminths are free living. So Planaria is free living. So is this guy right here in this image. And then some of them are parasitic. They might be ectoparasites, like the Monogenea. And they might be internal parasites like the trematodes and the cestodes. Okay, so let's take a look at those two classes at their life cycle. They're pretty interesting to look at. So the first one, the trematodes, which again we call them flukes for short. You may have heard of schistosomiasis. This is caused by a blood fluke. It causes organ damage and chronic fatigue. And this organism is going to infect about 200 million people per year, usually in tropical regions. And again, in regions of the world where there's not good sanitation. Okay, so let's talk about how this happens. So you've got two hosts in this life cycle. The primary host is the human, but there also needs to be a secondary host for this organism to complete its life cycle. And so the secondary host is usually, for schistosomiasis, is usually a snail. Okay, so what happens? Let's start here. So actually, no, let's start here. So we have this human, okay, and in this human we have the blood fluke. So we've got, there's actually two worms in here, and the worms are going to produce offspring, and those offspring will pass through the digestive tract of the human and come out in fecal matter. Okay, and so you end up with a larval stage of this flatworm, and that fecal matter has got to be in a region where there's snails present. Okay, so again, if you've got good sanitation measures, that won't happen, hopefully. But in countries that do not have those capabilities, third world countries, then oftentimes, you have human waste, fecal matter, in areas that it really shouldn't be. And so if that happens and you have fecal matter that contains larval forms of this organism, that can be picked up by the snail. And then inside of the snail, there's some developmental processes that happen to the organism. And then the snail will release another larval form here. And that larval form, if a human is in the water where the snail is, as well as the fecal material, then this guy right here, this is called the Cercadia larva, can actually enter into the human. It actually bores a hole through the skin. Okay, and once inside, it will develop into the adult form of the worm. And so you can see that if there's proper sanitation and proper hygiene, this could all be avoided. But unfortunately, in some parts of the world, again, this is infecting 200 million people a year, that's not the case. And so again, once this guy is inside, it's going to cause organ damage, and it will also cause chronic fatigue. Okay, so that's, again, the disease is called schistosomiasis, and it's caused by this fluke, this flatworm. Okay, the second class here that I just want to briefly mention, tapeworms. Tapeworms are, they're pretty neat to look at. Tapeworms are going to, again, be internal parasites. And again, they have a complex life cycle, and so in multiple hosts. So the primary host here again is the human, and then we have pigs or cattle as secondary hosts. And there's different species of tapeworm that will end up in these hosts. So for cattle, it's Tinea saginata. And in pig, it's Tinea solium. So they are species specific. Both of those tapeworms, though, can infect humans. Okay, so if we look at the head region of the tapeworm, so again, these do have an anterior end, a head region. They've got these really cool structures. The head of a tapeworm is called its scolex, and the head region usually has hooks and suckers that it will use for attachment to the host. And Tinea species tend to hang out in they'll be found in the digestive tract and the intestine of humans. And when you look at the adult version of this, so there's a head region here, and then you see this repeating structure, and this guy's actually not considered to be segmented, which is kind of interesting, even though it looks very segmented. If you look at each of these structures, if we were to kind of blow that up and look here, what we find is all of this, these are called proglottids, by the way, all of this is all reproductive structure. So both egg and sperm are produced, and these pieces actually break off in the host's digestive tract. And so these proglottids, which again are just chock full of reproductive structures, they will end up in fecal matter. Okay, and you get fertilization. So eggs can fertilize sperm, or sorry, sperm can fertilize eggs, and so you have fertilized eggs that will end up in the environment. Okay, so again, if there's not proper sanitation, then you end up with fecal matter from humans in the same region where you have pigs and cattle grazing. Okay, not a great idea, but it happens. If that happens, and the pig or the cow eats vegetation that has these eggs on it, well, then the eggs will be ingested, and they will actually penetrate the intestinal wall of these organisms and end up in the organism's muscle, where it develops into a larval stage that actually lives inside the muscle of these animals. Okay, and so then humans, well, we eat pig and cattle, and if we eat undercooked or raw meat from an organism that's infected, well, then this embryo will end up in our digestive tract, and that can then develop into the adult. Okay, and the cycle can continue. Tapeworms are really interesting. They don't have any digestive system whatsoever. And so they're inside of us being bathed in nutrients in our digestive tracts. And so what they do is they just steal our nutrition. Okay? And that's how they end up getting their food. Okay? Pretty nasty. Okay, so that's it for the platyhelminths. The next group that we're going to talk about, the next phylum, is the phylum Mollusca. Now these guys are mostly mostly found in marine habitats. However, some do inhabit freshwater environments, as well as they can be found on land, so terrestrial environments as well. It's a pretty diverse phylum. It has over 75,000 species. It's the second most diverse next to arthropods. And they have a very varied morphology, and it depends on the species that you're looking at. However, they do share some common characteristics. So all of them will have a muscular foot that's used in locomotion. They will all have a visceral mass, so a concentration of internal organs, and they all have a structure called the mantle. Some will have a shell and some do not. Okay, if they do have a shell, that shell is made out of material called calcium carbonate. Okay, so you actually saw an organism from the phylum Mollusca in lab. If you remember back, you saw Helix. He was a snail, right? A shelled organism belonging to the phylum Mollusca. And we're actually showing something that looks kind of like Helix down here. So what do we find? Again, we find a muscular foot that's used for locomotion. Again, it's going to vary in shape. It depends on who we're looking at. In the case of this snail here, the muscular foot is on the bottom surface here. Okay, that's the foot. But you'll see when we look at bivalves, the foot's going to look really different. Okay, in shelled mollusks, like bivalves or the snail here, the foot is retractable. It can actually be retracted back up into the shell. The visceral mass, so the organs, I gotta get the pen here. Okay. The organs, or the visceral mass, that concentration of organs, is found above the foot. So if our foot is here, here's the concentration of organs, all in here. Okay, we call it the visceral mass. So what do we have in there? We have digestive structures. We have elements of a nervous system. We have excretory structures. We have organs involved in reproduction. And we also have respiratory structures here. Here you can see the gill. Okay, that's involved in respiration. You can also see that this organism has a heart. Okay, you can also see nerve cords, right? Concentration of nervous tissue here, primitive brain. Reproductive structures here. So you see gonads; those are reproductive structures. Digestive structures. You've got your mouth here. Crop, right? Stomach, digestive glands, intestine, and then out here is the anus. You also have a coelom, a fluid-filled chamber. So these are true coelomates. This guy has gills. So usually aquatic mollusks will have gills, and terrestrial mollusks will have other structures that are similar to lungs. They may contain, this guy has it, a structure called the radula. The radula is a structure used in feeding. Not all of them will contain that. All mollusks are going to be multicellular, bilateral, triploblastic animals that have a true coelom. They have a true anterior and posterior end, as well as a ventral and dorsal surface, and right and left sides. Okay, so as far as reproduction, there are separate male and female organisms, and fertilization usually occurs externally. Okay, however, for some terrestrial organisms and for the cephalopods, one of the classes, reproduction or fertilization actually happens internally. Okay, we'll mention it when we get to those. These guys do go through metamorphosis, so they will go through larval stages before the young adult stage and then the mature adult stage. There are 7 classes. We're going to only go over a couple of them. As you can see, this chapter is pretty long. So I chose just 3 to cover. So the bivalves, the gastropods, and the cephalopods. And we're just going to briefly go over these 3 classes. So let's start with the bivalves. So when you hear the term bivalve, these are going to be organisms that have 2 shells hinged. Okay, you've heard of these. You've heard of clams. Maybe you've eaten them. Oysters, mussels, scallops. All of those are bivalves. So they're enclosed in a 2-shell hinged structure. They're found mainly in marine environments, and some are found in freshwater. Again, that hinge, where those 2 shells are connected, is going to be a dorsal hinge. Okay, and that hinge is going to be composed of ligaments. So those are the structures actually holding those two shells together. These guys, they don't have a true head region. It's kind of poorly developed. It's kind of hard to tell where that region is. They may have specialized sensory structures like eye spots or statocysts, okay? And feeding, they're going to lack that radula, that structure found in the mouth, kind of like teeth. Instead, they are suspension feeders. So they're going to actually have a structure called the incurrent siphon that's going to bring water in. Along with it, it'll bring any food that's in that water. And then they're going to kind of, their digestive structures will filter out the food, and then the water will exit. Okay, so here's a structure of what a typical bivalve would look like. You may have dissected one of these if in high school biology. They're a little hard to dissect. It's kind of hard to tell what's what inside of these sometimes. We only have one shell being shown here. There will be another shell here on top. And if you look here at the dorsal side, that's where you're going to have those ligaments that hinge those two shells together. And on the ventral side, that's where the shells are allowed to open up. And this guy will put out its muscular foot. Okay, there's the foot. And it's going to use that foot for locomotion. So it'll actually stick this thing out. It'll use it to move, and then it will pull that back in. And again, there's videos on YouTube that you can go on and watch showing some of that movement. It's kind of neat to watch. Lots of structures in here. So for filter feeding, here's the incurrent siphon, which is going to bring water in. Okay. And here's the excurrent siphon where water is going to leave. And we have structures here, for example, these gills, which are going to pull oxygen out of that water. Okay, so oxygen will be used by all these cells for cellular respiration, right? And we have other structures, for example, let's get some digestive structures in here. Some of it's hidden underneath here, but here's the stomach, right? We've got the esophagus here, the mouth right here, okay? What else should we be concerned with? We do have an intestine as well as an anus. Okay, and so again, that water comes in, food's going to be filtered out, taken up by the mouth, digestion happens in the stomach. We've got this intestine, and then any waste products leave via the anus. What else? We do have a circulatory system, so we have a heart-like structure, which is going to be involved in pumping. What else? We have metanephridium. These are kind of like kidney-like structures. Mantle shell. Yeah, we have nervous tissue, so ganglion. We talked about most of these as well. Okay, so yeah, so we definitely have our respiratory structures. We have digestive structures. We have circulatory structures. We have structures involved in locomotion, excretory structures. Oh, gonads, I missed that. So reproductive structures as well. Okay. Okay, so that's it for the class bivalve. Just wanted to introduce you to what they look like. So the next class is the gastropoda. Okay, this includes snails, slugs, and conchs. So again, what you saw in lab helix, that was a snail. Some of these guys will bear shells, so snails will bear shells, and then some do not. Okay, they are going to be bilateral, having a head and tail region, but sometimes they might be asymmetrical, especially the coiled part up in the shell. And the visceral mass, again, is going to be above the muscular foot. And it's kind of interesting. These guys go through something called torsion. Hold on one second. During development, that torsion is twisting. And so what happens is the visceral mass gets kind of twisted in here. And what we end up with is this is the anus, and this is the mouth. It's kind of weird. So the anus is above the mouth. It's kind of a weird positioning of the digestive structures. Muscular foot is on the bottom here, again used for movement. Some will have a very well-developed head region. Some of them will have tentacles, and some of them even have eyes. Okay, so all of these structures in here are all sensory structures, so nervous tissue. Okay, so the last class that I just want to briefly mention are the cephalopoda. This includes octopi, squids, cuttlefish, and nautilus. Ooh. Lost the page here. Hold on. Yeah, here we go. Okay. So in this picture here on the right, let's see, this guy's the octopus. Okay. Ooh. Okay, I'm having trouble with the pen. Here we go, sorry. So this guy right here is the octopus. He's pretty neat. And again, there's lots of YouTube videos that you can watch on all of these animals. Octopus are really smart animals. They can actually change color. And some of that is here, right? Yeah, vivid coloration, often used to camouflage. Some can change colors. And again, you can find videos of these guys actually changing colors. It's pretty neat to watch. So that's the octopus here. This is the cuttlefish. This is the nautilus. And this is the squid. So pretty neat. Some are shell bearing. If they do have a shell, it's usually internal. Okay, not all of them do, but some of them do. Again, very vivid coloration. Some of them can change coloration. And oftentimes that coloration is used as a camouflage to either hide them from something or to hide them if they're looking for prey, so they can kind of sneak up on them. They are active hunters, and they're carnivores, predators. They have beak-like jaws. Pretty neat to watch them. Again, lots of videos that you can go on and actually watch them hunting. They're very well studied. They have well-developed nervous system, and they have complex eyes. And they have a closed circulatory system as well. So pretty advanced organisms. Okay. The foot here is modified into tentacles. So that, again, is going to be the structure that's used for locomotion. And they can actually move by something called jet propulsion. They can actually push water, and actually that will speed up their movement. They do have separate male and female sexes. Fertilization is internal, and so mating does take place. Females will then lay fertilized eggs, which will then develop into new organisms. Some species will also produce dark ink. This is in response usually to being preyed upon. Okay, again, another mechanism to kind of hide them or confuse the predator so that they can get away. Hold on. Okay, sorry. So that was it for the phylum Mollusca. Our next phylum is the phylum Annelida, or the annelids. These are going to be still in the Lophotrochozoa. They are the segmented worms. They are going to be protostomes, right? Lophotrochozoa, and they are bilateral. And so again, they're going to have a right and left side. These guys are found in a lot of different environments. So they can be found in marine environments, they can be found in freshwater environments, and they can also be found on land in terrestrial environments. However, in these terrestrial environments, the environment needs to be moist. Okay. Some species are parasitic, some are free living, and some are symbiotic. So they'll form relationships with other organisms. There's about 16,500 species here of annelids. They include your common earthworm, which you're going to watch a video of a dissection this week of the earthworm. And you will also be completing some activities around that. And you can go out right in your backyard, dig up a little dirt, and you'll find tons of earthworms. They're extremely important organisms to soil ecology. Pretty neat to watch. The polychaete worms, as well as leeches, and again, some of these you saw in lab last week, are all part of this phylum. Some are hermaphrodites, where they have both female as well as male reproductive structures, and others are not. It depends on the species. There is a structure called the clitellum that is a common feature, and again, you're going to see that on the earthworm this week. It's used in reproduction. It's going to generate mucus, and it will aid in the transfer of sperm from one worm to another. So even though they're hermaphrodites, they don't self-fertilize. Okay, so there's actually a transfer of sperm from one organism to another, or one worm to another. So here he is, right? This is the common earthworm. And earthworms, they're pretty neat to watch, and they range in size. So, the ones that we have here, not too big, but you can actually go on YouTube or look up images, and you can actually see that there's giant earthworms in other parts of the world. Okay, so a common feature to all annelids is that their epidermis, their outermost layer of cells, is covered by a structure called the cuticle. Okay, and the cuticle, and so the cuticle has a couple of different jobs. It's really there for protection. Protection, number one, from drying out, desiccation, and number two, this earthworm is crawling through dirt, and there can be rough, maybe sharp objects. So it also is there to serve as to protect the epidermis. Okay, so it protects the earthworm from drying out, and it also protects the epidermis from friction. What else do we find? We're going to find on all annelids these chitinous, which chitin is a structural polysaccharide, so kind of hard structures, hair-like extensions on every segment of the body. These are segmented worms that are going to be called setae. Okay, and so shown here, it looks like here's a pair, and here's a pair, and again, you're going to see them on the earthworm. Those setae are involved in locomotion. Okay. They do have a true coelom, so they are true coelomates. They have a fluid-filled body cavity. And here in this picture, you can see all the space, all the empty space around these internal organs. That's all part of the coelom. They also have some pretty well-developed organ systems. So they have a digestive system with a mouth as well as an anus, as well as these other structures. So a pharynx, esophagus, crop, and gizzard. And all of that leads to the intestine, as well as the anus. And again, you're going to see all of those structures in lab. What else does it have? It also has, here's some more stuff that it has, another picture here. Before I get into some other structures, let me just mention a few other things. So this would be the anterior or head region where the mouth is located. Okay, and if we go down the length of this guy, this would be the posterior end where the anus is located. These little black things along the sides, really on the ventral surface of the organism, those are all setae. And notice how you can actually see segmentation on the outside of the worm. So this worm is segmented for sure. And they do display both external as well as internal segmentation. So you can actually see on the body wall on the inside that you can see segmentation as well. So definitely a segmented worm. Look at this structure right here, the clitellum it's called. That's going to be a structure used in sexual reproduction. Again, it's going to secrete mucus, which is going to aid in sperm transfer. And you'll see this structure in lab as well. Okay, so what else do we have here? So again, a complete digestive system. So let me point out some of those structures. So here's the mouth, right, at the most anterior end. It's kind of down on the ventral surface, right, of the worm. If we go back a little bit, here is the first pouch-like structure called the pharynx, okay, which is kind of equivalent to like our throat. And then so food will enter through the mouth, through the pharynx, through the esophagus, okay. That's the tube leading into the crop. The crop is where food is stored before it enters the gizzard. The gizzard is an interesting structure. If you were to break this open, what you would find is there would be some rocks in there. So the gizzard is actually a grinding compartment. And so soil, right, is going to be kind of ground up in here, and food, particles that are inside of that soil, it's going to be ground up and broken up so that there's access to that food. And then that's going to pass into the intestine, right, where absorption of that food will happen. And then all the other stuff, the dirt, right, what's left, is going to pass through and come out the other end. So think about how important that is to soil ecology. These guys, they aerate, they turn over the soil. It's pretty neat. And then their waste products also end up back in the soil. So they're really, really important organisms. What else do we have? Let's look at the circulatory system. So they have a closed circulatory system. If we look here, do you see these red swollen pouch-like structures? There's 5 of them. In this figure, we're calling them the aortic arches. They're also referred to as hearts. Okay, so there's actually 5 structures here that are responsible for pumping blood. And that blood will be pumped out. Is this labeled? Yeah. And it's going to travel through the dorsal blood vessel, which runs along the top of the organism's body. And you'll notice you've got all these connections here. And that is going to deliver nutrients throughout the organism's body. And then to bring that blood back to the heart, we have our ventral vessel. So that's going to run on the belly side of the organism. And that's going to bring blood, right, nutrient poor, oxygen poor blood back to these hearts. Okay, kind of interesting. What else do we have? We have some muscles. So the longitudinal and circular muscles, along with the coelom, are and the setae, are going to allow these worms to move. Okay. What else do we have? We have nervous tissue. So right here, shown in blue, there's a pair here of cerebral ganglion. That's the brain, the primitive brain. And then running along the whole length of the worm, you've got this ventral nerve cord. Okay, that's going to run along the whole length of the worm there. Am I missing anything? Not shown on this slide. I think it may have been on the previous slide. There will also be specialized excretory system called nephridia. Nephridia are going to filter out any impurities in the coelomic fluid, and they're going to get rid of that. So let me actually back up. Yeah, so you can see these structures here. These are nephridia, and they actually open up to the outside of the body wall. So they're involved in getting rid of waste. Okay. Okay. And in lab, you're also going to see through the dissection reproductive structures. So, the clitellum is involved in reproduction, but there's also going to be structures located at the anterior end of the organism that are responsible for the production of sperm and egg, and then sperm storage. So you'll see some of those structures. Some of them are very hard to see. Like for example, the testes, they're very, very hard to see in a dissection. So you won't see all of those structures, but you will see some. Okay, so here is the third. Are you with me? So again, you want to pause this and go ahead and answer those questions.
Okay, so before we go on, a couple of questions for you. This was kind of a long section, so there's kind of a lot of questions here. Okay, so the first one. All of the following describe most members of the phylum Platyhelminths except So are the platyhelminths, are they bilateral? Are they coelomates? Do they have a ladder-type nervous system? Do they have an incomplete digestive system? So take a minute to think about that. So which of these is not true? Okay, so hopefully you chose blue, coelomates. So platyhelminths are acoelomates. They are flatworms. So if we were to cut them and look at them in cross-section, we would not find a fluid-filled chamber completely lined with mesoderm. So they are not going to be considered to be coelomates. They are bilateral. They are triploblastic. They do have a nervous system that's in the form of a ladder, and they do have a complete digestive system with mouth, other structures leading to the intestinal tract, which leads out to the anus. Okay, so they do have a complete digestive tract. Next question. The head of members of the class Cestota, containing suckers and hooks for attachment to the host, is called the, what's that head called? Is it called the proboscis? Is it called the scolex? Is it called the proglottid? Or is it called the radula? Which one of those is the name of the head in the class Cestota? Okay, again, pause me if you need to look back or if you need more time. That head is going to be called the scolex. Okay, and again, it's going to have hooks as well as suckers for attachment to the host. These are parasitic worms. Okay, so again, blue, the scolex. The other terms on here, for example, the proboscis, that describes a muscular pharynx-like structure that's used for taking in food in organisms like planaria. It is not the head of the Cestota. The proglottid is one of the repeating units of a tapeworm, but it is not the head. Remember, the proglottids are going to be structures that are loaded with gametes, okay, with reproductive structures and reproductive cells, not involved in attachment. And the radula is not a structure found in the Cestota. Instead, that is found in mollusks, and that's going to be used in feeding. So the only one here that makes sense is blue scolex. Okay, here's some more questions. All of the following are true of most members of the phylum Mollusca, except they are triploblastic, they are radially symmetrical, they have a muscular foot, and they are coelomates. So which one of those is not true for members of the phylum Mollusca? Okay, so all of those are true except for blue. Members of the phylum Mollusca are bilaterally symmetrical, not radially symmetrical. Okay, so blue would be the only one that is false. They are triploblastic. They do have a muscular foot, which is sometimes reduced in some classes, and they are true coelomates. Okay, and again, that coelom might be modified as well, but it's still there. Okay, next question. Members of this class of mollusks are carnivorous predators with well-developed nervous systems, closed circulatory systems, and vivid coloration that is used in camouflage. So which one of these describes that class of mollusks? The cephalopoda, the gastropoda, the bivalvia, or the trematoda? So take a second to think about that one. Okay, so hopefully you answered cephalopoda. Okay, the cephalopods. Those are going to include organisms, again, that have all of these characteristics here. So you want to think about octopus, nautilus, squid, those structures. Those organisms all belong to the class Cephalopoda of the phylum Mollusca. Okay, so they are mollusks, that's their phylum, and they are cephalopods, that's going to be their class. Okay, the gastropoda include organisms like snails. Okay, the bivalves are going to include organisms like clams and scallops. Okay, and then the trematodes, those we talked about when we were looking at the platyhelminths. Okay, so those are flatworms, so those don't even fit the description at all. They're not mollusks. So the three mollusks that are here again are the cephalopoda, the gastropoda, and the bivalvia. And the only one that has all of these features, especially that last one, vivid coloration used as camouflage, that's going to be the cephalopoda. Okay, here's our last two questions. Members of the phylum Annelida have all of the following characteristics except. So are members of the phylum Annelida, are they bilaterally symmetrical? Do they have a coelom, right, a fluid-filled body cavity? Do they have an incomplete digestive system? Do they have a closed circulatory system? So which one of these is not going to be true? Okay, so hopefully you answered incomplete digestive system. These guys have a complete digestive system from mouth to anus. The rest of these are true. And hopefully you've looked at lab for this week. In lab, you'll be reading all about one member of the phylum Annelida, the earthworm. And you're going to be watching a video on the dissection, as well as answering lots of questions of labeling and other questions. So it goes really nicely with this part of the lecture. So yeah, so the Annelida are definitely bilaterally symmetrical. They have a right and left side when cut sagittally. They also show cephalization, right, the development of a head region that has a concentration of nervous tissue, right? These guys are going to have structures called ganglia or brains. Most will also have nervous tissue running from that concentration, so they'll have some type of a nerve cord. They are true coelomates, having a fluid-filled body cavity completely lined with mesoderm. And they do have closed circulatory systems. And again, you're going to see, and we looked at a picture a few minutes ago, but you'll see in lab as well. With our example of the earthworm, they have a closed circulatory system, including 5 pseudohearts, and they have both a dorsal as well as ventral blood vessel. Okay, so definitely a closed circulatory system. So the only one, again, that is not true for members of the phylum Annelida is in green. Their digestive system is complete. Okay. Okay, last question here. The structure that covers the epidermis and helps keep annelids moist is that called setae, or the cuticle, parapodia, or the pharynx? Okay, so hopefully you answered blue, the cuticle. Okay, the cuticle is going to be a structure that's going to be found outside of the epidermis. It's going to cover the organism's entire body. It varies in thickness depending on which species or class of organism that we're looking at. But again, the role of that cuticle is to prevent desiccation, which means to prevent the organism from drying out, or in other terms, to keep the organism moist. Okay, so that's the last question here. Again, you'll do the next section, then I'll have some more questions for you.
Okay, we're getting close here. So we've covered the flatworms, the mollusks, and the annelids. All of those belonged to the Lophotrochozoa clade of the protostomes. Ooh. Okay, again, those were bilateral eumetazoan animals. So now we're going to go into the other clade, the other group of protostomes here. and I always butcher this word, the Adixozoa. That's better. Okay. And we're going to talk about just some of these. We're not going to talk about all of them, but we'll talk about some examples from a couple of the phylum. Okay, so the two phylum that we're going to concentrate on are the nematodes, or the nematoda, which are the roundworms. And you saw an example of the nematodes last week in lab. Ascaris was a roundworm. And you saw a bunch of examples of the arthropoda. I think you saw the crayfish and the grasshopper and the tick. Probably some more that I'm missing. There was a bunch of them. The arthropoda are going to be the most abundant and diverse. It's the most diverse phylum. When we look at the arthropoda, we're going to see that its members have a tough, and the nematodes, the members have a tough external covering called the cuticle. The arthropoda have an exoskeleton. This has a couple of different jobs. It's going to prevent desiccation. It provides protection. However, this structure must be shed as they grow, and so all members of this superphylum are going to molt, or molting is also called, I can never say this word, ecdysis. And so all of these are going to go through this process of molting, shedding their exoskeleton. And that exoskeleton gets replaced. Okay. Okay, so let's talk about the nematodes first. And look, this looks just like Ascaris that you saw in lab. So the nematodes, which again are more commonly referred to as roundworms, again, these guys are triploblastic and bilateral. They are also pseudocoelomates. So they have a fluid-filled body cavity, but it's not completely lined with mesoderm, so a little bit different than a true coelom. Some are free-living, some are parasitic, and we find them in all habitats. So there's aquatic, freshwater, marine, as well as terrestrial roundworms. In cross-section, they're round, and so that's where they get their name. And these guys are not segmented, so they do not show external or internal segmentation. They do have a complete digestive tract with a mouth and anus, so one opening where food enters and the other where waste is excreted. They do have a nervous system composed of a ganglion in the head region, as well as nerve cords extending the length of the organism's body. Not the latter type, but they do have nerve cords. They have a dorsal as well as a ventral. So just like we saw in the earthworm, in the annelids. Okay, they also have reproductive structures, and they are covered by a cuticle. Okay. Again, some of these worms are parasitic, so we're going to talk about that in just a second. Fertilization for these worms is internal, and these guys do go through larval stages, so they do go through metamorphosis. And the roundworms, again, they live in all environments, but they can be parasitic. And so in this particular example, this is a life cycle showing the parasitic worm. In this case, in this particular example, this is the guinea worm. The guinea worm infects about 3.5 million people a year in Africa specifically. And so if we look at this life cycle, it's a little bit small, kind of hard to read, but you have a human, right, who drinks unfiltered water that contains the larval stage of the guinea worm. After drinking it, the larvae end up in the digestive tract, and they penetrate the host stomach and intestinal wall, and they end up maturing and then reproducing inside of those structures. Okay. Then the fertilized female worm migrates to the surface of the human skin, causes blistering, and the larvae are discharged out of those blisters. Okay. Larvae then get released into water, and the female worm, I can't read this very good. It begins to emerge from the skin 1 year after infection. Okay, so the worm, that's actually shown up here. The worm is actually starting to come out of the skin of the human. That's kind of gross, right? It looks like they've wound that worm around something here. Obviously, it's coming from a blister. Okay, so how do you get this? You're drinking unfiltered water, right? And then that worm ends up in the digestive tract. Reproduction happens. The worm travels through the human's body to the skin, forming a blister. And then the female worm actually discharges the eggs right out of that blister, and she will also appear at the skin surface. Okay. Larvae now in the water. They are consumed by another organism, a copepod, which is shown here. So there is a secondary host here, a very complex life cycle, just like we saw with the other parasitic flatworms. And then that larva is going to go through some different stages, and eventually will be shed from the organism, and again ends up inside of water. And then, you know, this person is drinking water where larval form of this worm was shed, and they end up ingesting that larval form. So again, really complex life cycle. But you can see how sanitation would fix this. But again, in certain parts of the world, there is not clean drinking water. And so, you know, people are going to drink water from places where maybe they also, you know, bathe. They're actually inside of the water where they also drink water from. Okay, very dangerous. Okay, phylum Arthropoda. This is the most successful phylum. These guys are segmented. They also have jointed appendages or legs. Any structure coming off of the body is an appendage, and they're going to be jointed. 85% of all of the animal species on the planet belong to this phylum, so it is the most successful phylum. These guys have an exoskeleton. They go through molting of that exoskeleton, so they shed it and they have to regrow it. They have a true coelom, and again, they are protostomes, just like the nematodes. They are also bilaterally symmetrical, triploblastic, and these guys are also found in all habitats. There are 5 subphylum, and we're going to talk about just a couple of these. So there are the trilobites, which are extinct. They're no longer found on the planet. However, we have beautiful fossils of them. There are the hexapoda. These are the insects. There's the myriapoda. These are the millipedes and centipedes. There's the crustaceans and the chelicerae, which include the arachnids, so spiders, as well as scorpions. Okay, common feature to all species within the phylum Arthropoda, they all have an open circulatory system. So they do have heart or hearts, but they're not going to pump into vessels. Instead, there's going to be an open blood cavity called the hemocoel. Okay, so they do have a circulatory system, but it's open, not closed. Respiratory systems vary. Many will have a trachea and these structures called book lungs where gas exchange happens. Aquatic animals will have gills. They also have complex excretory structures to get rid of waste products. Okay, here's an example of an organism from the subphylum Hexapoda. So the subphylum Hexapoda, these are going to be arthropods that have 6 legs, or we could say 3 pairs of legs. These guys are segmented. They have specific regions or segments of their body. So they're going to have a head segment, then they have a thorax, and then they have an abdomen. So 3 separate segments. When we look at the thorax region, we're going to find the attachment of 2 wings. And again, the thorax region is going to also have extensions coming off, jointed appendages coming off, or legs. It includes all classes of insects, and this is going to be the largest class. What we're going to find as a common feature for all insects is on the head region, there's going to be one pair of sensory structures called the antenna. In the mouth region, there's going to be mandibles. These are mouth parts. And there's going to be one pair of compound eyes, pretty complex compound eyes. When we look at the abdomen is going to hold the structures used for reproduction. And so you can see the bee here in this picture. Let's look at the structures here. So again, we have three segments. We have the head region, and in the head region, we're going to have the concentration of nervous tissue, as well as sensory organs like the antenna here, as well as, you can kind of see this, the eye, right, the compound eye. There will also be a nerve cord, a dorsal nerve cord, okay, extending down both the thorax and the abdominal region. If we look at the thorax region, again, this region is going to have parts of the digestive system, parts of the circulatory system, parts of the nervous system, as well as two sets here of legs, okay, as well as wings. Yeah, so we also have wings attached to the body at the thorax region. And then the abdominal region down here, again, we have a concentration of this, in this case, this is a female, so reproductive structures. Okay. Okay. The second subphylum, Myriapoda. Again, these are arthropods. Look at these guys. These are probably the worst things that I find these in my basements all the time, right? Centipedes. Not fun to look at. Creepy crawly, really fast movers. These guys don't just have 3 sets of legs or 6 legs. Instead, they have numerous legs. So somewhere from 10 to 750 legs, up to 750 legs can be found on these guys. The most common organisms that, again, you're probably familiar with are millipedes and centipedes. All of them are terrestrial, so they're all land-dwelling organisms, but they like to be in moist, humid environments. We find them in moist soils, or decaying organic matter, or, you know, in your basement. Okay. The next subphylum, and again, we're just kind of briefly looking at these just to take a survey of some of these. The next subphylum is the subphylum Crustacea. This is going to include organisms that are found in marine, freshwater, and terrestrial environments. You've heard of these before, right? So you've all probably eaten shrimp, maybe lobster, crabs like the guy here in this figure, crayfish, pill bugs, roly-polies. These are all examples of Crustacea. And again, you saw some of them in the lab last week. Okay, when we look at organisms that belong to this subphylum, Crustacea, we find 2 pairs of antennae on their anterior end of their body, so their head region. They also, they have mandibles as part of their mouth parts. And they also have biramus, or 2-branched appendages. So if you look here, here's an appendage, and you can see that there's actually 2 appendages kind of branched off here. So that's called a biramus appendage. So that's going to be a common feature in all crustaceans, Crustacea. They also have a cephalothorax and an open circulatory system that again has that hemocoel, or that blood cavity, as well as a heart. Okay. Okay. So, okay. Our last subphylum here, the Chelocerata. This includes some organisms that you're probably familiar with, like spiders, scorpions, horseshoe crabs, and sea spiders. Again, you saw spiders, a spider, in the lab, okay? Phylum Arthropoda, subphylum here. They are mostly found in terrestrial environments. However, there are some aquatic, so sea spiders are definitely aquatic. Horseshoe crabs are aquatic. They as well have a cephalothorax and an abdomen, so a fused head and thorax region and then a separate abdomen. Their first pair of appendages are these structures called chelicerae, which are claw-like mouth parts, or they might be fang-like, and these guys lack sensory antenna. Their second pair of appendages is used in feeding. They're called pedipalps. They also have an open circulatory system, which contains a heart and a hemocoel, again, a blood-filled cavity. If they are aquatic, they have gills. If they are terrestrial, then they have internal respiratory structures. They have a trachea or book lungs. Okay, so they have respiratory system, they have a circulatory system, they have a complete digestive tract, they do have sensory organs. Okay, they are covered by an exoskeleton, they have jointed appendages. Okay. Again, ooh, look at this guy. You've heard of him, right? Internal digestion, so they have complete digestive tracts. Digestion usually happens internally, although there are some species who display external digestion or are parasitic. They do have a nervous system composed of a brain and 2 nerve cords. Reproduction is done by external fertilization, or depending on the species, there are some that display internal fertilization. Some actually care for their young, and then some species do not. It depends on the species that we're looking at. Okay, so here is this Are You With Me? So you want to describe members of this group, including both phylums, the nematodes, and the arthropods. When you're finished, you can go on.
Okay, before you go on, a couple questions for you. So here's the first one. What does the term ecdysis mean? Is it a form of asexual reproduction? Does it mean to molt or shed? Does it mean having a complex life cycle? Or is it does it mean that organisms can live in both aquatic and terrestrial habitats? So which one of these is the definition for ecdysis? Okay, so hopefully you chose blue, to molt or shed. So remember, members of this group, of this clade, are going to undergo molting or shedding. Okay? Okay, next question. Which of the following is not a key advantage provided by the exoskeleton of terrestrial arthropods? So does the exoskeleton prevent desiccation? Remember, desiccation means to dry out. Does the exoskeleton protect internal tissues and organs? Does the exoskeleton provide mechanical support? Or lastly, so which one of these is not true? Does the exoskeleton grow with the arthropod throughout its life? So which one of these is not a key advantage? So hopefully you answered yellow. The rest of these definitely are advantages to organisms that have this exoskeleton. But in yellow, it says grows with the arthropod throughout its life. That is not true, right? As arthropods grow, they have to actually shed or molt. Remember that term ecdysis that we just talked about in that first question? They have to lose that exoskeleton and then secrete a new one. So again, in yellow, that's not true. The exoskeleton does not grow with the arthropod. Very different from internal endoskeletons that would grow with the organism. Okay? Okay, next question. Grasshoppers have 3 body parts, including a head, thorax, and abdomen. They have 6 jointed legs, 2 pairs of wings, 2 antennae, and are covered by a hard exoskeleton made of chitin. Okay, chitin is a polysaccharide. Okay, we also find chitin in the cell walls of fungi. Okay, very hard structurally supportive polysaccharide. Which phylum and subphylum do grasshoppers belong to? So again, read through those traits and try to figure out which phylum. So the three phylums that are listed here, are they arthropods, annelids, or nematodes? Okay, so that's the first part of the question. And the second part, after you choose which phylum, which subphylum? So if they're arthropods, are they of the subphylum Hexapoda? If they're annelids, are they of the subphylum Hexapoda? If they're arthropods, are they of the subphylum Chelicerata? Or if they're nematodes, are they of the subphylum Myriapoda? So which one of those is correct? Okay, so grasshoppers are going to belong to the phylum Arthropoda, they're arthropods, and they're going to belong to the subphylum Hexapoda. Okay? The example, if we go back a couple slides, was the bee, remember? Okay, so again, just like the bee, it's going to have three body parts: the head region, the thorax, and the abdomen, unlike others which have different parts. Pair of antennae, pair of wings. Okay, and we look at the jointed appendages here. There are 6. Okay, so again, that's going to be Hexapoda. Okay. Last question, I think. Yeah, last question. You find a terrestrial, which means land-dwelling organism, that exhibits bilateral symmetry, has an exoskeleton, and has upwards of 100 jointed legs. Which subphylum does this organism belong to? Would this organism belong to the Hexapoda, the Chelicerata, the Myriapoda, or the Crustacea? Which subphylum would this terrestrial organism belong to, with over 100 jointed legs? Okay, so hopefully you answered green, the Myriapoda. Okay, that's going to be the subphylum where we would place this organism into. It's an arthropod, and it's going to belong to the subphylum Myriapoda. Okay, that's the last question for this section. We've got one more section to go, and I'll be back on with some more questions.
Okay, we're getting close. So our last grouping here contains the deuterostomes. And so the deuterostomes, remember, includes 2 phylum that we're going to talk about. There's 3 phylum, but 2 that we're going to talk about: the echinoderms and the chordates. Okay, now both of these display bilateral symmetry, and they are in the deuterostome clade. So when we look at development, they have radial cleavage, and that cleavage is indeterminate. And they display enterocoely for the development of the coelom. Okay, so a couple of features there. Okay, so let's talk about the echinoderms. Again, you saw some examples, or a example, I should say, of this in the lab last week, and then we have the dissection of the starfish this week. So common features for all echinoderms is that they all possess spiny skin. And so these guys actually have an endoskeleton, and the endoskeleton kind of protrudes up through the epidermis, and so we end up with these spines. Okay, and those spines, again, are going to be found on the external surface of the organism. They are marine animals, so they're found in aquatic marine environments. And they include organisms that you're probably familiar with, like sea stars and sea cucumbers, and the one that you saw in lab was the sea urchin, sand dollars, and brittle stars. All of those belong to this phylum. Now, they're placed with chordates in the deuterostome, and they're placed, therefore, with bilateral animals. And so this is kind of strange. So here's the thing. So the larval stage of these organisms, they do go through metamorphosis. The larval stage is bilateral. Okay? The adult stage, however, is usually radial. And in the case of sea stars and brittle stars, they're pentaradial. Okay, in the case of sea urchins and sand dollars, those guys are just radial. Okay, so why are they placed with the bilateral animals? Again, because the larval, this should say stage, not tage, is bilateral. Okay, again, they have an internal skeleton, the endoskeleton. Okay, hard substance. On their underside, which we're going to call the oral surface because that's where the mouth is located, they are going to have down each of their arms, if it's a sea star, they're going to have rows of these things called tube feet. Okay, and those tube feet are going to aid in attachment to surfaces. They're also going to aid in movement, and also not on here, they also aid in feeding. Okay. They do have a coelom, but it's modified and it forms a structure called the water vascular system, which is a system that's going to be used in movement, locomotion, along with those tube feet. Okay, so here's a nice little picture right from your textbook showing some external as well as internal structures. And again, you're going to dissect this in lab and you'll be answering some questions about its structure and some labeling. These guys do have a simple nervous system, which is actually pretty impossible to see in the dissection. Here in this figure or in models, it's much easier to see what the structure looks like. The nervous system is going to consist of a structure called the nerve ring, okay, which is on its closest to the mouth. Okay, so here, and I'm not even sure if they're showing it on here. They're not. It would be down here, so it would be a ring. It actually kind of looks like a hexagon, but it's in this region. It's in the central, this part right here of the starfish is called the central disc. And that ring, that nerve ring, is going to be found here. And then there's going to be a nerve that's going to extend down each arm. Okay, and again, it's not showed on here. They're showing some other structures. Okay, so each one of these here is an arm, right? And you can actually see the spiny skin. You can see those little white structures coming up. And then again, this region here is called the central disc. And there's a concentration of nervous tissue here, as well as digestive tissue, okay, concentrated here in this region. Okay, reproduction. Echinoderms are going to reproduce sexually. They are going to release gametes, so egg and sperm, into the water. Fertilization is external. And there will be separate sexes. So starfish either produce egg or they produce sperm. And where is that produced? You can see this structure here called gonads. That is going to be where egg and sperm are produced. And so in the dissection, it kind of looks like this gooey stuff down each arm underneath the digestive structures. So in this arm here, this has been less dissected. If we took out these digestive structures, we'll talk about those in a second. If we cut this right here, that's what this looks like. So we cut that. And so we can actually see what's underneath those digestive structures. And one thing that's there are the gonads, which again are going to be the gamete producing structures. They can reproduce asexually by fragmentation. They can regenerate body parts. So they're pretty neat. Okay. I just want to mention feeding a little bit. So in the dissection, you'll see a lot of these structures. So, and if you get a chance to go, on YouTube and type in, starfish eating a clam, it's kind of neat to watch. So these guys are predators, and so they're gonna, and they're carnivores. So they're gonna find their prey, maybe a bivalve like a clam. They're gonna actually hold that with their arms, with their tube feet. So these little structures here are gonna run down the length on the oral side, the mouth side, of each of these arms. And these are very flexible. They can bend their arms, and the tube feet can actually suction, kind of grabbing onto whatever they're about to eat. And their arms can actually pull, unhinge a bivalve. So they can open it up, and they only have to open it up just a little bit, because what happens next is pretty neat. So what's going to happen is this guy has a couple of stomachs, 2 different stomachs, and the stomach on the bottom will actually be released out of the mouth, will enter into whatever the organism is eating. So in this case, if it's a bivalve, it enters into the bivalve. Digestive juices get released. Digestion actually happens externally for the most part, and that stomach kind of gathers up that soup of nutrients and internalizes it back into the mouth, and then digestion is finished inside the organism's body. So it's pretty neat. And again, you can watch that. Go onto like a YouTube video and try to watch that if you can. So the digestive enzymes, the digestive juices that get secreted, they're all made in these digestive glands, which are called the hepatic ceca. And again, you're going to see that in the lab. And then this little duct right here called the hepatic duct allows those digestive juices to enter into the stomachs, which are in this region. Digestion happens, nutrients get distributed throughout the organism's body, and then any waste products will exit out the top or the aboral surface through this structure here called the anus. I also quickly just want to mention the water vascular system. I told you that it was involved in movement. I just want to point out a couple of other structures in here that, again, you're going to see in lab in the dissection. So on the aboral surface, so the non-mouth surface where the anus is, near the anus, there's going to be a larger, more prominent structure. It looks like a button, and it's called the madreporite. The madreporite is going to be a structure in which water enters into. My mouse is being funny. So water enters into the madreporite. It then travels down this structure here called the stone canal, and it eventually enters into this gray ring called the ring canal. And then that water is going to be pushed down each arm. This is called the radial canal. And that water is going to control the tube feet. There's a little bulb at the top called the ampulla. And if the water pushes onto this, that ampulla is going to push the tube foot out. And then when water isn't being pushed, this will be retracted in. So the water vascular system controls whether the tube feet are out or in. When they're in, they're suctioned. When they're out, they're not suctioned. And again, they're going to be used for movement. So these tube feet are not only used for feeding, but they're also used for locomotion. And again, the water vascular system is going to be the structure that supports that movement. Okay, there's 5 classes of echinoderms, and I just want to mention them here to you. The first class you're probably all very familiar with. It is the class Asteroidea, or the sea stars. That's their common name. The second class, these guys are really cool to watch. These guys are fast. These are called the brittle stars. You saw one of these guys. This is a sea urchin from the class Echinoidea. This was one of the animals that you had to classify last week in the lab. And then if we come down here, this is I think this is a sea lily. Okay, belongs to the class Crinoidea. And then the last class here, these guys are really cool, sea cucumbers. As you can see, we're already at 2 hours for this lecture, so I couldn't put in everything that I really wanted to. It's just too much information. But if you have a chance to check out sea cucumbers, these guys are so cool. One of their defense mechanisms when they are being attacked is to start to discharge, actually body parts, out of their anus, which is really neat to read about and to actually watch them in action. They're pretty cool. Okay, the last phylum that I'm just going to introduce you to, again, we're running up on time here, and we can't get too deep into it. Is the phylum Chordata. Okay, 4 key features to this phylum. First off, they have a notochord. So remember Amphioxus from lab? A notochord is going to be a cartilaginous skeletal rod that's there to support the body. It will be there in all embryonic and some adult chordates. Okay. Some will have a dorsal hollow nerve cord, and that will develop into nervous tissue. All of them have pharyngeal slits, at least at some point in their life cycle. Okay. And they all have a post-anal tail, at least at some point in their life cycle. There's 2 clades of the invertebrate chordates. They are the urochordates, or the tunicates. These guys live on the ocean floor and they're suspension feeders. And then the one that you saw in lab, cephalochordate, the lanceolate, that's where the amphioxus is. Those guys are suspension feeders, but they're not sessile. All other chordates are vertebrates, which again, vertebrates are discussed in the next chapter. Okay, so again, I didn't give you too much information about the invertebrate chordata, but again, we're running up on time. So for your last Are You With Me, you're just going to describe the key features of the deuterostomes, tell me a little bit about the echinoderms, and tell me just a little bit about the chordata.
Before we completely wrap up this chapter, we got a couple questions for you. So the first one, just 2 questions for you. The first one, adult echinoderms do they have triangular symmetry, radial symmetry, hexagonal symmetry, or pentaradial symmetry? So hopefully you answered yellow. They show pentaradial symmetry. However, if you answered radial symmetry, you might have an argument there. The better answer is pentaradial symmetry for the adult echinoderms. Remember that the larval stage is bilateral, and that's why we group them or classify them as having bilateral symmetry. But again, as adults, they're going to be pentaradial. Last question here. Which of the following features does not distinguish humans as a member of the phylum Chordata? Okay, so which one of these is not a distinguishing feature of humans as being part of this phylum Chordata? So in pink, human embryos undergo indeterminate cleavage. Blue, a spinal cord runs along an adult's dorsal side. Green, human embryos exhibit pharyngeal arches and gill slits. Or yellow, the human coccyx forms an embryonic tail. So which one does not distinguish humans? It's not a distinguishing feature. So what the question's really asking you is which one of these is not a distinguishing feature of the phylum Chordata? So think about that for a minute. Okay, so first off, all of these are true about humans. Okay, human embryos do undergo indeterminate cleavage. Humans do have a spinal cord that runs along the adult's dorsal side. Human embryos do have pharyngeal arches and gill slits. In humans, the coccyx does form from the embryonic tail. Okay, so those are all true. However, one of them is not a distinguishing feature, and that's the first one, pink, because all deuterostomes undergo indeterminate cleavage. So do you see how it's not a distinguishing feature? So if we were to look at the echinoderms as well as members of the phylum Chordata, all of them are going to display indeterminate cleavage during those early embryonic developmental stages. Okay, and so that's not a distinguishing feature. The rest of these, so blue, green, and yellow, those are all distinguishing features. Those make members of the phylum Chordata different from members of the phylum Echinodermata. Okay, that was a little bit tricky. All right, so that's the last question. And so you want to make sure that your Are You With Me's are complete. You should have already uploaded your vocab list, your chapter outline. You want to get your Are You With Me's now uploaded to Blackboard. Take some time and study, and then you're going to go ahead and you're going to take the quiz for chapter 28. Okay, and in lab, make sure that you get all of the analyte assignment completed as well.